I'm Marta Illescas. I'm a virology specialist and a PhD student in the San Cecilio University Hospital. And first of all, I would like to express my gratitude to the scientific committee for having selected this communication for its oral presentation. I would like to start mentioning the importance in the accurate diagnosis and knowledge of HDV, considering that HDV prevalence, although HDV prevalence remains to be fully characterized, between 12 and 70 million of people are believed to be infected with this virus worldwide. Furthermore, this virus exhibits a high genetic variability, having been described 8 different genotypes, each enclosing multiple subgenotypes. A genetic variability of 16% within the same genotype and among 20 and 40% between different genotypes can be found. And the genotype 3 is the most genetically distant of all. Genotyping, it's important due to the genotype may influence the disease progression, the treatment outcome, and the diagnostic accuracy. Relating its epidemiology, genotype 1 is the most prevalent genotype worldwide, followed by far by HDV5. And although genotype 1 has a wide range of clinical presentations, some studies had found it to be associated with worse prognosis and a high risk to progression than genotypes 2 and 5. Furthermore, a worse treatment response has been observed in some cohorts in contrast with genotype 5. On the other hand, genotype 3 has been associated with outbreaks of fulminant hepatitis in specific area of the Amazonian basin. So a comprehensive characterization of the virus requires a deeper understanding of its genetic diversity and clinical implications. However, in Spain, molecular characterization of the virus has been limited over the years, making it difficult to determine the currently circulating genotypes. In fact, the only available study in that sense is dating back 1998. So our main objective was to characterize the molecular epidemiology of HDV in Spain through whole genome sequencing. Our study was divided in 2 distinct phases from August 2019 to June 2024. The first phase of our study was conducted genotyping samples from Andalusia, Murcia, and the Canary Islands. And from July 2024 onward, genotyping was extended and samples from Galicia and Extremadura were also included in the analysis. Genotyping was performed in patients with HDV RNA positivity confirmed through whole genome sequencing. Before the whole genome sequencing, a thermal shock strategy was employed with the objective of relax the supercoiling of HDV RNA and enhance the RNA amplification. This strategy consisted of heating at 95 degrees during 10 minutes and a subsequent freezing for another 10 minutes. After that, an overlapping primer strategy that has been following a protocol that has been used before in our laboratory was employed to whole genome amplification. An Illumina segmentation indexing protocol was used on an XZ 1000 for whole genome sequencing. CLC genomic workbench was used to sequence assembly and HDV database for genotype determination. The phylogenetic relationships were inferred using neighbor joining method, including sequences with plus than 90% coverage in Mega software. And finally, the recombinance events were assessed using the RDB5 60.63 software. A total of 151 patients were included in our study with an average age of 50 years. The majority of them were males, and the median HDV RNA level was almost logarithm 5. The 52% of patients were of Spanish origin, and the second most prevalent place of origin was Eastern Europe, with 13% of patients coming from Romania. Relating the genotyping results, a total of 144 samples were successfully genotyped, representing the 95% of cases. The average coverage was of 98%, and the most frequently identified genotype in our study was genotype 1 with the 88% of cases, followed by genotype 5, identified in the 10% of cases, highlighting that this genotype was only found in patients from West Africa. Furthermore, one case belonged to genotype 7 and another one to genotype 3. The phylogenetic analysis was performed in a total of 138 samples with plus than 90% coverage. Within genotype 1, the most frequently subgenotype was 1D with 87% of cases, whereas in case of genotype 5, the most frequently identified subgenotype was 5B. None of the subgenotype sequences revealed evidence of recombination. So in conclusion, HDV genotyping plays a key role in understanding its molecular epidemiology in Spain. According to prior research, genotype 1 remains the predominant genotype among Spanish patients, whereas genotype 5 is only found in individuals from West African countries. So these results provide important insights into the genetic diversity of HDV in Spain. Finally, I would like to acknowledge to the technical and medical staff of the Clínico San Cecilio University Hospital, especially to Drs. Federico García and Adolfo de Salazar for having supervised this work, and to all the collaborating centers. Thank you.